Combination therapies for the treatment of chronic lymphocytic leukemia are currently under development, and there is many trials who really try to address this problem. There is a recent report, although not really fully done, or at least it's not published in an abstract form, but at least in a press release, that the combination with ibrutinib with a monoclonal antibody of obinutuzumab can produce a significant progression-free survival in comparison with obinutuzumab chlorambucil alone, something that is pretty much similar to what we have shown in other trials. In this case, it's important to consider that ibrutinib is given for a long period of time until disease progression of unacceptable toxicity, and those patients has a very low rate of progression. So this combination is still unclear how they're going to pan out in the introduction of the change of paradigm in the treatment of CLL. However, there is other combinations with ibrutinib and a BCL2 inhibitor, venetoclax, that really is showing very remarkable responses in terms of complete responses, and even more absence of minimal residual disease by flow cytometry in the bone marrow. MRD negative in the bone marrow is becoming, with the introduction of venetoclax, a goal to be achieved in patients with chronic lymphocytic leukemia. However, MRD negativity, although is very well studied for the last years after chemoimmunotherapy, is not incorporated in any guidelines, no IWCLL or NCCN guidelines, but in the future may be a goal to achieve in patients treated with these more complex combinations such as ibrutinib and venetoclax, with the goal to have duration times of therapy and to be able to stop therapy and wait until patients progress, or in this case, to really have a long period of time without any treatment. There is today several combinations that are being studied for the treatment of frontline or even relapsed refractory CLL. Most of these combinations really entail the use of BCR inhibitor in the form of BTK inhibitors such as ibrutinib, or even other drugs such as acalabrutinib or other second-generation BTK inhibitors. Otherwise, the use of PI3K delta or gamma delta inhibitors is another area of investigation, as well alone or in combination with monoclonal antibodies. Once again, these strategies really continue to deliver the oral drug until disease progression of unacceptable toxicity. However, a second strategy and a second combination is the introduction of venetoclax, a BCL2 inhibitor, with a BCR inhibitor. In general, any of the BCR inhibitor available, BTK or PI3K. And even more recently, we have seen combination with the triple monoclonal antibody anti-CD20 BCR inhibitor, mostly in the form of BTK inhibitors such as ibrutinib, and a BCL2 inhibitor as venetoclax. Lastly, the German group is now working in several trials, trying to stratify and sequence several therapies, such as induction with a very short chemotherapy with bendamustine, followed by all the combinations I just discussed briefly a second ago, such as monoclonal antibody with a PI3K, monoclonal antibody with a BTK inhibitor, and as well the use of different monoclonal antibodies anti-CD20.